Hello, everyone. I'll introduce the paper submitted to IMETA. The title is, Short-Term Probiotic Supplementation Affects the Diversity, Genetics, Growth, and Interactions of the Native Gut Microbiome. The human gut harbors a highly diverse and dynamic microbiome that plays a crucial role in maintaining overall health and physiological homeostasis. Probiotics are a promising strategy for supporting gut health. Both preclinical and clinical studies have demonstrated the potential of probiotic supplementation in managing various health conditions. However, the effects of probiotic can vary significantly among individuals. And the precise mechanisms through which probiotics interact with and reshape the native gut microbiota, especially at the species and genetic levels, remain underexplored. Therefore, this study intervened with high-dose probiotic probiotic-mediated in healthy individuals and conducted deep metagenomic sequencing on multiple fecal samples to investigate the impact of the probiotic on gut microbiota from both macro and micro diversity perspectives. In this study, we selected a well-characterized probiotic strain, probiotic-mediated, which has been demonstrated positive effects on gut health, and administered it at a high dose of 200 billion CFU per day in a short-term intervention study involving healthy individuals. We performed deep metagenomic sequencing on multiple fecal samples to investigate the impact of the probiotic on gut microbiota. Firstly, by analyzing the diversity of gut microbiota, it was found that following the administration of probiotic-mediated, both diversity indices showed a significant increase, demonstrating that the intervention effectively enhanced the gut microbiota diversity. Notably, Bifidobacterium animalis demonstrated a significant increase in abundance at day 7 compared to baseline period. Analysis using average nucleotide identity confirmed that the increased abundance was primarily due to the administered probiotic-mediated strain. To further understand the subtle genetic shifts within the gut microbiota that may be driven by external factors, the single nucleotide variation in intestinal microflora was analyzed. Our findings revealed a non-significant increase in the number of species harboring SNVs following probiotic-mediated administration at day 7 compared to the base period. And SNV loci were classified as shared or non-shared, based on whether more than 50% of the loci differed between pre- and post-intervention. Among them, 2,442 non-shared loci were distributed across 120 genes, 21 of which were associated with carbohydrate transport and metabolism. The in situ bacterial replication rate was assessed using the IREP metric. An IREP value closer to 2 signifies a higher replication rate. Our analysis revealed that the replication rate of different strains was different after probiotic consumption. Interestingly, upon analyzing Bifidobacterium animalis specifically, we observed an increased replication rate following probiotic-mediated consumption, with the IREP value almost reaching 2 at 14D. Therefore, probiotics can influence the growth dynamics of the indigenous gut microbiota. Analysis of the dominant species revealed that their relative abundance changed significantly after the intervention, but no significant differences were observed in the NSNV across these species at any time points. Therefore, while the probiotic-mediated intervention led to notable ecological shifts within the gut microbial community, these changes were not accompanied by significant genetic adaptations within the dominant species over the study period. Spearman correlation analyses was used to evaluate the changes in interbacterial relationships over time, focusing on shifts in interaction strength and patterns. We found that probiotic-mediated exhibited the highest number of associations with other resident gut microbes at day 7, indicating an expansion of its interaction network following the intervention. Quantification of the nodes and edges in the network centered around probiotic-mediated showed that both metrics peaked at day 7 but declined subsequently during follow-up, suggesting that the enhanced connectivity was transient. Finally, it is concluded that short-term administration of probiotic-mediated enhanced gut microbial diversity and influenced key microbial properties, including nucleotide variation, growth dynamics, and interbacterial interactions. And the microbial community appears to undergo rapid and multifaceted ecological adjustments, potentially preceding longer-term evolutionary changes.